All right, now let's talk about section 3 from chapter 14. Um, some of the I want to go through some of the techniques we have used to study the human genome and other genomes for that matter, just genomes in general. Um, so there are these uh, enzymes called restriction enzymes that allow us to cut up DNA. All right. Um, And so that ability to cut DNA, and then we're going to talk about this technique called electrophoresis, where we can separate pieces of DNA. And then there are techniques we can use to do DNA replication outside of cells and test tubes, basically. So these three things have really allowed us to do a lot of things in studying genomes and manipulating genomes, in particular genes, and moving them around, and essentially has enhanced the field of biotechnology and allowed for um, genetic engineering to occur. Um, so first of all, restriction enzymes. These are enzymes that are naturally found in bacteria, and they basically cut DNA. Bacteria use them as a defensive mechanism because there are viruses that attempt to infect bacteria, and they inject the viral DNA, and the bacteria use these enzymes to sort of attempt to ward off that foreign viral DNA. Well, we figured out that we can use these enzymes from these bacteria to cut any kind of DNA. Um, and the different enzymes get names that are basically abbreviations of the bacteria they come from. For example, EcoR1 is a common one, and it comes from E. coli, and that stands for E. coli restriction enzyme 1. And so these restriction enzymes cut at particular sequences of DNA, as you can see here. So for example, ALU1 always cuts where it says AGCT or and TCGA on the opposite strands. And you notice it cuts straight across, and the same with this one here. Now these other ones, BAMH1, HIND3, EcoR1, they all cut in a zigzag pattern, okay? But again, they always cut at a particular sequence, so EcoR1 will cut wherever there's a GAATTC. And where you have these sequences in DNA, these are known as restriction sites. This, wherever you have a GAATTC, would be an EcoR1 restriction site. And that's where that enzyme would cut the DNA. Now notice when you cut in a zigzag pattern, what happens is you leave what are called sticky ends, that is unpaired bases on the ends of the piece of DNA you have cut. These sticky sites are very useful when we want to move DNA around and splice one piece of DNA into another piece of DNA, as we'll discuss in a moment. So why cut up the DNA? Well, first of all, you can take a very large piece of DNA, essentially the DNA in a whole chromosome, and cut it up into smaller pieces, which are more manageable, easier to work with. Um, and as we'll again see, you can then move this DNA around. um, Cutting up DNA is also useful for studying diversity because not every individual has the same restriction sites. That is, you can find these places where the restriction enzyme will cut the DNA, and it can vary amongst us. And so again, these restriction sites can vary, and so this studying of the variation using restriction enzymes is what's known as RFLP analysis. This stands for restriction fragment length polymorphism. That is, because the restriction sites can vary, when you cut different individuals' pieces of DNA, the DNA pieces can be of different sizes because of the difference in the restriction sites. And so that, thus the term restriction fragment length polymorphism. Um, and so, as I said previously, again, so these sticky ends are quite useful because they allow for the moving of DNA um, between individuals, even between species, and for the process of genetic engineering. So, as this example shows, you may have this piece of DNA, and this DNA would go on in this way and this way, but you notice there's this EcoR1 restriction site here. Well, if we take a piece of DNA from some other individual or even some other species that has also been cut with the EcoR1 restriction enzyme, notice the sticky ends match up, and so you can splice one piece of DNA into another, again, because those restriction enzymes match up, and you create what you would call recombinant DNA. You have recombined two pieces of DNA. And this is a very common thing that's done in genetic engineering. I'll give you a good example here. So insulin, you've probably heard of that. It's a protein that is used to control blood sugar levels in mammals. And people who are diabetic, they have the gene that's responsible for making this protein is defective in some way, and so the insulin they don't may not make enough of it, or the insulin they make is not quite working right, and so they often have to get insulin shots. Well, we used to have to get insulin from basically dead animals, cattle, sheep, horses, pigs, things like that, when they were slaughtered, basically for food. And so this insulin was rather expensive, and it was a very laborious process to get it. Um, but what was done several decades ago is that bacteria, essentially E. coli, had the human gene, the human insulin gene, spliced into them, and so now there are these bacterial types, these cultures of bacteria with the insulin gene, this human gene, and they sit there all day and they pump out insulin. And so now these pharmaceutical companies have these cultures of these genetically engineered bacteria, and they're able to produce an abundant supply of insulin much more cheaply than before. Um, so that just kind of shows how DNA is DNA, and once you move it around, the organism doesn't care where it came from. It's the same stuff as their DNA. It's again a sequence of A's, T's, C's, and G's, and so you put the gene for insulin into a bacteria, and that bacteria will start pumping out insulin. Um, all right, so now we go to the separation. So now, when you have pieces of DNA or DNA that you've cut with restriction enzymes, you'll get all these smaller pieces. And a technique you can use to then separate those pieces is what's called gel electrophoresis. And I'm going to show you the stuff. We have some of the apparatus for doing this, and I'll show it to you in class later. Um, and it'll probably be clear. What you do is you have a gel, just like Jell-O, gelatin, and so a gel is basically a matrix of some kind of polysaccharide, typically, um, that encloses a liquid, water, pretty much. And so it sort of the polysaccharide holds the liquid inside this matrix, and so it forms what we call a gel. So it's kind of a combination of a solid and a liquid, or sort of held together by the solid part. Well, what we do with our an agarose is a typical polysaccharide that comes from seaweed that's used to make the gels we use in gel electrophoresis. Um, so what you do is you've got your sample of DNA that has been cut up into small pieces. And some pieces are larger than others. Some of the pieces are really small, some are kind of large, and some are in between. And what you do is you load them into the gel, and I'll show you how this is done. And you run a current through the gel, and you load your DNA up at the negative end, or what's called the cathode, and the DNA will run to the positive end because DNA is a negatively charged molecule, so it will go towards the positive electrode with the current. Um, and so you load your DNA in the gel, you run it for 30 minutes, 40 minutes, 50 minutes, an hour, however long it takes, and the pieces of DNA move down the gel. Which ones move down fastest? Well, the smaller ones do because they're smaller and they can get through the gel easier. The bigger pieces move more slowly and they don't go as far down the gel. So you're able to separate the pieces based on their size. Um, Typically, you add a sample of known DNA of various sizes so that you can see approximately how big the pieces are that you have separated out. Um, it's possible to then stain that gel, and then you can literally see where the pieces are in the gel. And then if one wants, you can isolate, you can basically remove those pieces from the gel if necessary. Um, so this process of creating restriction fragment lengths using restriction enzymes, separating those pieces on a gel using gel electrophoresis, that is the basis of what you call DNA fingerprinting, a common technique used to identify people or identify the remains of people in accident cases, or if there's a homicide or something like that. I'm sure they talk about it in these crime scene dramas on TV. Um, because when you look at parts of our genome that are quite variable between us, the restriction fragment lengths that you get between 82 people will most likely be different. Um, and so it's again a way to identify individuals or things you get from a crime scene. If there's a murder and you find blood at the crime scene, and of course some of it is from the victim, but let's say the perpetrator also was cut and left some blood behind, you can isolate DNA from their blood and basically generate a DNA fingerprint and then use that when you have suspects to see if it matches. And if it matches, it's pretty solid evidence that that individual was there and was most likely the perpetrator. All right, another technique, DNA sequencing. Um, so here we are literally figuring out the sequence of A's, T's, C's, and G's in that individual's, um, perhaps their complete genome or just an individual gene. You can do the whole thing or just parts. So you're figuring out the sequence of bases. Um, and this is useful to identify particular mutations they might have at a very detailed label. It's useful to compare the variation between individuals, the variation between species. When you compare the sequence of DNA in chimps versus people, they're very similar to each other. There are differences, of course, but they're very similar. Of course, if you compare any two people, they're very similar, but there will always be some differences between us. So there was I'm going to stop here on this video and complete section 3 in the next video because I'm running out of time. We'll talk about the Human Genome Project on the next video.